Thank you, Tim. I appreciate it. So my name is John Deaton, and I will start the presentation in presentation mode. There we go. I'm Vice President of Science and Technology here at Deerland, Probiotics and Enzymes. Our main headquarters is in the United States. We're actually in Kennesaw, Georgia, just north of Atlanta. We actually have two facilities at that location, one an enzyme facility, and then the other one a probiotic facility that handles spores as well as non-spores. We are a supplement company. We formulate with many different probiotics and enzymes. And we develop our own innovative technologies as well. Where there's gaps in the market, we develop probiotic, prebiotic, enzyme-based formulations. And we have several different locations that we do research, as well as just facilities. We have several in the United States, but we also have others internationally, both in Denmark as well as Ireland. We do a lot of research and
sorry, John, just to disturb you there for 1 second. I don't think your slides are moving on. They may not. If you've got two screens, that might be the problem, or it may not be in presentation mode.
Let me try again.
You can scroll through them like that if you want to. There's no problem there.
Okay, just make it easier. Sorry about that.
No problem.
We actually have several locations, several in the United States, others in Denmark and Ireland, where we do a lot of research. We do a lot of collaborations with universities and other institutions. We basically we research everything from in vitro to in vivo. Our purpose is actually to find solutions, supplemental solutions to help people in their daily lives, feel better, be able to do more, be able to be more active, etc. All of our technologies are around probiotics and enzymes, of course, and then we, of course, are responsible for the customization. We do the product technology as well as the education, both with our customers as well as the end consumers. We are a B2B company, but we do research that ends up educating the consumers in terms of how our products can help them. One of our products, one of the things that we've looked at for a long time is a enzyme-based antioxidant formulation. Free radicals are everywhere. They damage your tissues, skin, internal organs, etc. It's part of the aging process. It gives you wrinkles and basically can cause various issues as you get older. And so we wanted to have a very efficacious product. We wanted it to be enzyme driven so that you can have a small amount that gives you a strong activity. And so that's the reason that we actually pulled from chloroplast. So we use spinach, we use an extract, we use enzymes to isolate that. We remove the cell wall because your body can't digest that. We also do certain techniques to actually stabilize as well as enhance the chloroplast contents so that we keep the glutathione pathway, which is what we're most interested in, intact. And so we can detect all these components in it. And then, of course, the glutathione pathway is Mother Nature's best and strongest antioxidant pathway. Every organism has it. And it helps eliminate free radicals in many different forms and areas, tissues, and liquids, as well as handling the different forms, hydroxyls, peroxyls, singlets, and superoxides. And then, but one of the things is, of course, we want to show the how it actually helps directly. We know that you get a lot of free radicals from many different areas, from stress, pollution, poor diet, etc. But we really wanted to narrow this down and actually show real life situations. And that's where we turned to Shannon ABC and collaborated with them on a project for this product. And I'll turn it over to Shannon to go through that.
That's great. Thanks very much, John. So if you can take your presentation off share, and then Neil, if you want to start away there. And thanks very much, John, for setting the scene for that.
So can you see my screen okay?
Yeah.
Okay, thanks very much. And thanks, John. So my name is Neil Burke, and I'm a business development scientist and project manager working for Shannon ABC, primarily at MTU Kerry. And I'm just going to give you a brief overview of some of the R&D work that we carried out in collaboration with Deerland. So let's see if my slide will move. Okay, so a lot of the work that we carry out at MTU Kerry involves cell culture. And for those of you who mightn't be as familiar with that, it involves the removal and isolation of specific cell types from either an animal or a plant. And essentially then it means growing them artificially in the lab. So just to mention some of the advantages of using cell culture for R&D work. Firstly, they're an ethical and economical alternative to testing on humans or animals. And they're also very time efficient. So the cells typically tend to grow reasonably fast, and you can use them at scale. So it can be reasonably high throughput. As well as that, the cell culture approach allows the testing of multiple parameters and endpoints. And so you're getting a lot of detailed information for one experiment. And then I guess the real selling point of it is if someone has a product or wants to test a compound and their end target is a specific tissue, then this approach allows you to test that. So for example, if you're interested in looking for efficacy in muscle tissue, then we can isolate and work with muscle cells, and so you're getting specificity there. And so overall, cell culture is a dynamic biological model, and we use it routinely, as I mentioned. So just to give an example of an application for cell culture work, a company or a research product will have a research project even, sorry, would have a natural extract or an active ingredient of interest that they'd like to test. And so what we'll do is we'll take that and we'll bring it to these cultured cells. So each of these wells that are highlighted here would represent a population of a certain cell type. So for argument's sake, a muscle cell. And then you treat each individual well with a different concentration of your sample, and you'll have your respective and relevant controls in there as well. And from that, it allows us to measure a range of different endpoints. And just to mention a few, we can see the effect of extracts or compounds on the cells in relation to the cell viability, the cell growth or proliferation, how much free radicals or ROS is produced within the cell in response to it, or if it can reduce those levels, and if it can modulate things like inflammation or DNA damage. So in relation to our work with Deerland, John has mentioned the product Solarplast that they're looking at. And as you can probably remember from your school biology, a plant cell has a cell wall, and John mentioned earlier that it's difficult for that to be, the contents of the plant cell to be available when the cell wall is intact. And so we helped characterize the preparation of the Solarplast in order to remove the plant cell wall and to free up the protoplast and chloroplast that are within that, and they contain the bioactives of interest. So John mentioned about antioxidants and free radicals, and essentially what they do in the cell is they stabilize unstable compounds that can cause all sorts of cellular and tissue damage. And one in particular, as John highlighted, was the glutathione and the glutathione pathway. And as this was of a key interest to a Solarplast product, we were able to use a commercially available glutathione assay kit and test the levels of glutathione activity after the preparation of the Solarplast, excuse me. Okay, so from that point then we were able to focus on some of the key target tissues. And one of the applications for Solarplast is for skin health. And if it was potentially to be used as a topical product, you can see here in the diagram we have three kind of broadly categorized layers, common layers of the skin. And the outermost layer is known as the epidermis. And the epidermis is primarily made up of cells called keratinocytes. And so we have keratinocytes, and we're able to grow those keratinocytes in the lab and add our Solarplast to the keratinocytes. with different insults. So essentially we're stressing the cells and causing the cell to increase their amount of free radicals or ROS levels. And then using this dye, which you don't need to know the name of, it's things in science never tend to be tip off the tongue, but essentially once we add the dye to the cells, the dye is taken up by the cell, and the more ROS or free radicals that are present within the cell, the higher reading we get. So on the graph on the right is a representation of a treatment where you will see an increase in ROS. And so we were able to treat these cells, these skin cells, with an insult, as well as the product Solarplast, and we observed that Solarplast reduced the damage caused in relation to ROS and the free radicals. Again, so because Solarplast is, well, it has lots of beneficial attributes, as well as kind of improving skin health, it's also been implicated with gut health, as Deerland have done a lot of work around this already. And so what we were able to do was to take the Solarplast and process it through an artificial digestion protocol. And so essentially we're trying to mimic what would happen if a consumer or customer is ingesting the product. And so what we do here is we subject the Solarplast to the pH and the typical enzymes and salts that will be present at each stage of ingestion and digestion. So we go through from when it's in the mouth and how the saliva acts on it, to when it goes into the stomach and then on into the intestines. And at that point then, after we processed it, artificially digested the product, we were able to test to see if it had retained its antioxidant capabilities. And so we got a readout here, and there's an example on the right of the different levels of antioxidant capabilities that you can have from different samples and well-known antioxidant sources. And so we then confirmed that after the artificial digestion, Solarplast retained its antioxidant capabilities. And then again, just to kind of add to that information, another beneficial attribute of Solarplast is that it can help the gut barrier. So we use a CACO2 cell line, which is a commonly used intestinal epithelial cell line. And so that can mimic what's happening in the intestines. And what we do is we grow the cells, as you see from left to right here. We grow them individually, and we develop them or differentiate them into a more tightly formed tissue, for want of a better expression. So it forms an intact layer, and it mimics the barrier that's present in the gut. And then it's a similar approach to what we have done in other assays. We can treat these cells with an insult, and we can then try and prevent the damage caused by adding in Solarplast. And we can do that using electrical resistance. So we have a specialized instrument that passes an electrical current through the layer of cells, and essentially the more intact the cells are, the more their electrical resistance will be recorded or measured. And so if the cells are damaged or disrupted, you get a drop off in that reading. And so we're able to use that approach to show that Solarplast can protect that barrier after an insult. So just to conclude, we've characterized the optimal Solarplast preparation in relation to the chloroplast for efficacy testing. We validated the retention of Solarplast bioactivity after the different preparation protocols and the artificial digestion protocol. And we confirmed its antioxidant activity in both the skin and gut setting, and demonstrated cell barrier protection in the gut as well. And so all of this information really strengthens the existing data that Deerland have for their Solarplast product. So thanks very much for listening.